Hello and welcome to this video on the isoelectric point. This is the pH measurement that corresponds to a molecule having or not having free electron charge. This is important in protein chemistry, cell biology and more. You may have seen it listed as PI, pH parentheses I, or IEP. The isoelectric point is important in biochemistry and biology due to the effect it has on a wide range of cellular functions and mechanisms, protein shapes, and more. The isoelectric point of a molecule is the sum of its charges. Each chemical arm, such as an amine or carboxyl group, carries a charge that is negative, neutral, or positive. These groups are what give the molecule its isoelectric point, but also what is changed or acted on. For example, treating a cell with an acid can cause the cell wall to open in places, allowing egress and ingress. The same cell or cells can be washed or treated to counteract the change in the isoelectric point. A useful tool if you do not want to destroy a cell, but get something inside of it or get something out. As is obvious by now, the isoelectric point is a product of chemistry in each molecule. Depending on what you are measuring, there are different ways to calculate the pH. For proteins, this is relatively simple. With the isoelectric point established for proteins, as they become part of more complex structure, the ratio of positive, negative, and neutral units is used to calculate the dominant charge. Other methods use formulas to calculate the chemical groups. You will find examples of these calculations in the description box below. The example above of inserting foreign elements into a cell has been oversimplified to describe the changes involved. This change is achieved through a number of means, but the most common is the use of an acid or base as a solvent. The effect of this is to shift the isoelectric point of the sample. For example, proteins with a nominal negative charge at neutral pH would have a more negative charge in an alkaline solution, or a more positive charge in an acidic solution. If you have a bi-lipid cell wall, this creates a polarizing charge which pushes lipids apart and or away from the cell-embedded proteins. This creates cracks and gaps. The reason for this can be simplified to what follows. The chemical groups that make up the isoelectric measure have a larger percentage of elements that readily give or receive electrons. By placing them in an environment that favors the donation or receiving of electrons, these groups become more or less pronounced. When used with proteins, this process is called protonation or deprotonation. That will be covered in a coming video on how the isoelectric point is used. Shifting the charge of a molecule like this might seem strange, but it does have uses. Purification of samples, like the process used in HPLC, is an example. Similar principles are used in proofing DNA and RNA primers for amplification. You can see that here with gel electrophoresis. For this reason and others, many nutrient solutions, growth media, storage solutions, and more contain buffers. These are compounds which can fluctuate between a base and acid as needed. This keeps the largest solution it is part of in a narrow range of pH. This prevents any drastic changes to that which is being held in that solution, for example, protonation. A coming video will show you how this works in practice. Thank you for watching this video. Please post any comments, questions, or suggestions you may have below.